I am so pleased to be sitting here with one of my favorite people in the whole wide world, Dr. Francis Collins. Dr. Collins, you recently stepped down as Director of the National Institutes of Health. And as your farewell celebration highlighted, you have touched so many lives. You've touched my personal life, but lives also of other scientists at the NIH, the citizens of America, but also the citizens of this world. And just recently, you have been named as Acting Science Advisor to the President. For today's award, the John Edward Porter Legacy Award, generously supported by Ann Lurie, let's start with the word legacy. As a tie into that, now that maybe you have had some time to step back, breathe and rest, and reflect on your life and career so far, what are some of the moments that stay with you?
Oh my, that's quite an opening question, Kizzmekia. It's wonderful to be able to have this conversation with you. I will confess that I got asked, who would be the person you would most want to conduct this interview for Research America and this Legacy Award? And yeah, that's why you're there. So really glad that we can have this opportunity to talk about science and where it's been and where it's going. And you are certainly somebody who is a big part of where it's going with all that you've been doing in virology and immunology and all that you've done to make it possible for the vaccines for COVID-19 based on mRNA to be a reality that is saving millions of lives. So before we go any further, Dr. Corbett, thank you for your amazing contributions. And I know there's many more to come now that you're perched up there at the Harvard School of Public Health. Well, I am deeply honored to be recognized with this Legacy Award and just want to say thank you to Research America for this selection, which I certainly never expected. And, you know, I might also have to say it is a great honor to be part of a program that includes a lot of other people being recognized with awards who I am very much in awe of. People like Rosa DeLauro and Kay Granger are being recognized for their congressional role in support of NIH. People like my former colleague, Vivian Pinn. People like Jed Manicherian, who founded the Act for NIH effort that has made such a difference in getting visibility to why biomedical research is maybe one of the most important investments the federal government can make. So thank you, Jed, especially. And many others. If I go through the whole list, I'll use up all of our time. So you asked me a specific question. What are the moments that stay with me? You know, I guess this is a moment to kind of think back over the stretch of my experience as a biomedical researcher, which has just been an amazing adventure, maybe all the way back to my first opportunities as an independent investigator, trying to come up with solutions to problems in medicine that seemed like they needed attention. Back at the beginning of my efforts at the University of Michigan, deciding to take on really challenging, maybe unsolvable problems at the time about how can you identify a mutation in a gene that causes a genetic disease about which we know almost nothing except it's heritable. And a big effort that my lab put into that ultimately with a collaboration with Lapchi Choi in Toronto, finding the gene for cystic fibrosis in 1989. That was a moment, and it kind of opened the door to being able to do that for other diseases. Today, there's about 6,800 genetic diseases for which we know the specific DNA misspelling, which is pretty astounding considering where we were 30 years ago. I got to mention the Human Genome Project, the chance to lead that phenomenal group of 2,400 scientists. That's what it ultimately involved in 6 countries, all agreeing to put their shoulders to the same wheel and try to read out those 3 billion letters of the Human DNA Instruction Book at a time where it was really hard to do that. People listening to this, graduate students, are like, well, that's no big deal. I could do that myself in a couple of days. Well, it wasn't quite that easy back then. And to have that sequence emerge, to read out those letters, to see them appear, and to make sure that they got put on the internet every 24 hours with no intellectual property claims on them because it's our shared inheritance, that was just an experience of a lifetime. And now certainly since then, having the chance to serve for 12 years under 3 presidents as the NIH Director, amazing things have been possible. I mean, Kizzmekia, you would agree that this is a moment of phenomenal exponential opportunity in biomedical research. And to be able to do things like start the brain initiative that's going to figure out how our brains actually do what they do. and work on the cancer moonshot and put together this precision medicine program called All of Us. All of those were pretty amazing. Then, of course, we had this little interruption of this guy who arrived on the scene, SARS-CoV-2. And those years that I had the chance to try to bring together partnerships and figure out everything that we needed to do for vaccines, for therapeutics, for diagnostics, those were memorable, to put it mildly. And I must say, a willingness of scientists from every sector and every environment to just drop everything and jump in to try to see what they could do to save lives was inspiring. And the results of that, and Kizzmekia, you had a big part in this, namely having vaccines proven safe and 95% effective in 11 months by rigorous analysis, wildly outstripped the expectations that almost anybody would have put forward, and has made it possible for this terrible pandemic not to be nearly as bad. Although we still have this struggle, maybe we'll talk about that, about convincing the public that these are advances that everybody would want to take care of. So yeah, it's been a wild ride. And of course, I thought maybe I was going to have more time to reflect about this. This was supposed to be 2022, where I was going to have my equivalent of a sabbatical year to think deep thoughts, go back in the lab, which I love, do some writing. Well, that lasted less than 2 months. And now I, once again, I guess, have a public service gene that doesn't know when to turn off. And when asked to serve as the acting science advisor to the president, I could hardly say no. So got a new job, and I'm honored to be in that role, trying to do everything I can for our country, for our president, to steer the scientific mission.
Wow, as you go through all of those moments in your career, it's so inspirational. I remember when I first met you, I think I was maybe 19 after my first summer at the NIH, and you gave a presentation and everyone was talking about, how famous you were. And I felt that it was just so admirable that you were taking time out of your night. It was, I think, a 7 p.m. presentation for summer students to talk to us. And it was so inspiring how your career trajectory had gone. And as we think about inspiration for early career researchers like myself and those everywhere, what would you say would be the most useful piece of advice that you received in the beginning of your career?
Yeah, thinking back to that, maybe when I was a postdoc at Yale with Sherman Weissman was my postdoc mentor, doing partly clinical work and mostly learning molecular biology. I guess one of the things I learned from Sherman was, if you're going to spend your time doing research and it's going to be pretty demanding, you may as well pick a problem that's really important. Because, there's lots of ways you can carry out medical research, and there's a lot of really good, solid projects that will add knowledge to what we know about life and about disease. But if it's possible, pick something that might actually be more fundamentally significant, if it happens to work. That usually means you're stepping into a territory that's going to be higher risk. But if it succeeds, you've really made a contribution. So I know it's a little scary to a junior investigator just starting out to say, well, get out there and take a really big risk. And you might want to have a sort of backup plan of something that's a little more bread and butter, where you're going to be able to publish something. But mostly, I think it is your moment. You're the independent investigator. You get to decide at that point what you're going to work on. Pick on something that you could really be excited about. That's what I did at Michigan when I got there. I didn't publish a paper for the first 3 years as an assistant professor, and I was a little freaked out about that. But I think it was the right thing to do, ultimately. It was energizing. It ended up in this effort that was ultimately successful after another couple of years to find the cystic fibrosis gene. And isn't that what we ought to be thinking about right now? Because, as I said earlier, scientific opportunities are so exhilarating right now. Technology makes that possible, and working in teams makes that possible. That's the other thing I would say. Do not be shy to jump in with a team, because that's often where the really sparks will fly and things will happen, especially if you have people with different disciplinary expertise. So yeah, teams, and take a risk.
That's interesting, because one of my mentors actually gave me some similar advice about taking risk, but also working on a project that is essentially kind of the bread and butter and will certainly result in publication. And when we think about fear as we move in as early career investigators, as we move throughout our career, one of the things that I've also been told from people before is that, you know, part of the reason why science is so beautiful is because you can only fail upward. Even the failure gives us and the world actually knowledge. And so I like to kind of keep that in the back of my mind as I'm about only 6 months into starting my lab. So when we think about your career, when you think about your career to date, so we know about the advice that you've heeded along the way, but what are some of the things, or even just one thing that you feel like you would have done a little bit differently? And you can't say take naps. Because that's always my answer.
Okay, I'll skip that one. Well, there's probably plenty. I will say, when I first came to NIH to run the Human Genome Project, 1993, I really wasn't quite sure how to do this. I had not really run anything bigger than my research lab. And all of a sudden, it was this international effort to try to bring together lots of smart people, not all of whom had small egos, and figure out how they could work in a team-oriented way that would have explicit milestones and goals. And it took me quite a while to figure out how to do that. I think I was a bit too laissez-faire, just, let the flowers bloom and see what happens. And some of the flowers bloomed and some of them curled up and died. And there was needing to be a bit more gardening in that particular circumstance than I had initially had the sense of confidence to be able to do. So yeah, the first 2 or 3 years that I was involved in leading what was then the National Center for Human Genome Research, I was really kind of feeling my way. I think if I'd had more of a sense of confidence and vision, I could have done a better job in those first couple of years. Ultimately, you're in that position. You're either going to figure it out or you're going to get out of there, or somebody will get you out of there. It began to click, and I had lots of great advice from people who had done more of this, and a great team to work with. And ultimately, it ended up being an amazing experience, a coordinated effort. But it was a rough start.
Yeah, I think I hear so many scientists talk about their rough starts. All the scientists who are famous in their own rights and who have contributed so much to our scientific knowledge. When we think about contributions, one of the things that we think about regarding you in particular is how much you've contributed to our understanding of a simple term, genomics. Your work in this arena and so many others has been just remarkable. What is your vision for genomics in terms of helping society as we move forward?
Well, that's a great question. Yeah, I have been privileged, I think, with this role as leading the Genome Project to help shape where genomics is going. But man, it is just going everywhere without any help from me now, because it's such a powerful set of technologies and insights. And it's kind of looking at the center of the center in terms of biology. It's the instruction book that causes that cell or that tissue or that organism or that person to have a lot of the biological properties that we want to understand and that give us the opportunity to prevent or help treat disease. So it's very central now to everything we're doing, from newborn screening to cancer treatment. It's also becoming more and more clinically relevant in a big way. And I love that. As a physician, I always hoped that this would not just be an amazing academic adventure, but would also make differences at the clinic. And that's happening in a huge way. I don't know that I could have anticipated all of the ways in which this has played out. I don't think, for instance, that I would have imagined that a lot of the genomics that is now so exciting is really more about RNA than it is about DNA. It's being able at the single cell level to ask that cell what it's doing, the single cell biology. If you really want to query a human tissue, be it the immune system or, in my lab's case, the pancreatic islet, you want to know what every cell is up to. We weren't able to do that until just the last few years. And now genomics has stepped in with these very high throughput, high accuracy, low cost efforts to be able to look at that. And it's just a revelation of what you can see when you don't have to basically blur everything out by looking at an overall piece of tissue without understanding what's within. The cell is the basic unit of human life. We can now look at those cells and see what they're up to in a way that I would not have imagined. So omics, in every way you can think of, is making up a set of tools that everybody wants to use to get further understanding. Look what's just happened with the brain. This astounding set of publications just a few months ago, actually looking cell by cell what is happening in the prefrontal cortex of mice and monkeys and humans, and how can we really begin to understand the diversity of cell types, much greater than we actually knew from other less elegant methods, and beginning to reveal some of the complexity and some of the functional capacity of the brain in ways that are quite energizing and inspiring. And of course, where you want to go is to use that information to really begin to take apart things like schizophrenia and depression and PTSD and autism in ways that we desperately need to know more about. And that's happening. I keep coming back to this. This is such a moment of exponential scientific opportunity. I'm almost a little jealous, Kizzmekia, of you and others who are at your stage of starting out in their independent research programs, because you guys are going to have a blast.
Well, let me tell you, it always is so refreshing to hear scientists with, your type of clout to talk about how excited they are about the future of science. But I am equally as jealous because I would like a break too. We are all really burned out, I think, after a couple of years of this global pandemic. And while science has proved itself once again, I think that there are obviously some concerns with trust in science that may be waning. And as someone who is a trusted advisor to the president, has been the trusted leader of what is the world's most phenomenal biomedical research entity, what do you think about the waning of trust in science, and how can we as young and enthusiastic investigators and scientists kind of help in that regard?
Well, I'm glad you brought this up, because all of my excitement about progress of science has to be tempered by this issue, which has emerged so blatantly and in such a terribly concerning way with COVID. That is the diminution, it seems, of trust in science. I didn't dream back in 2020 when you and the entire community of vaccine developers were working night and day to come up with an effective prevention against SARS-CoV-2, that we would encounter such a big problem of resistance. I mean, here we are today, Kizzmekia, something like 50 million Americans have yet to get their first shot. And something like 100,000 plus Americans have died needlessly because they were talked into some kind of misinformation about the vaccines that made them fearful of taking advantage of it. In the most technologically advanced country in the world, for this to have happened is deeply troubling. And it does say that trust in science has somehow really slipped at just the worst possible time. It's hard to put all the reasons together. There's certainly a history here of perhaps a growing sense that scientists were a bit elitist, perhaps a bit separate from the common people on the street. Maybe a sense also that science had an agenda that was not entirely to the benefit of others. I don't think that's true for all of us who have devoted our lives to this, and most of us have not gotten rich as a result. I think this is an altruistic enterprise, but not everybody sees it that way. And politics has gotten into this in a really destructive way, which is heartbreaking. To have your political party be the strongest predictor of whether or not you've been vaccinated just makes absolutely zero sense. But that's the way it currently is. I think maybe, Kizzmekia, you and I tried our best in the course of the last 2 years, and you did a lot of this outreach, trying to explain the science of vaccine development and explain the science of what this virus was all about. And you did a lot of good, and I hope I did some good, but I think we underestimated the resistance, and maybe we should have had a deeper think about how to organize our science communication in a way that would have had a better chance of being heard. Because I think for a lot of people, just like don't want to hear it because it conflicted with some other messages that their particular tribal group had already brought on board. Almost a sort of like a litmus test. You can't be in our tribe unless you're opposed to vaccines. And that's hard then to get past that. So as an agenda going forward, I think we need a deep investigation into how science communication can be more effective in an area where people are intrinsically resistant. Maybe we are good at quoting statistics and not good enough at telling stories because people respond to stories. And scientists tend to say, well, that's just an anecdote. I can't use that as my argument, but it's what resonates.
Yeah. And during my time in this moment with the outreach that I've done, I think that one of the things that I realized too is that in our communications as scientists, sometimes we aren't necessarily listening. We're used to reading, but we're not necessarily used to listening, especially when people are not talking in a scientific language, which obviously the general public is. And so I think that we have to kind of add that to our arsenal of tools that we're going to use moving forward with our science communication. All right. Well, it's been a pleasure, Dr. Collins. I do want to end on one final question. So one of the things that you and I have in common is that we are fairly churchy. We consider ourselves to be Christian. And so my last and final question is, what is your favorite Bible verse and why?
Oh, well, I have many, and that's a great question. And yes, I am a person of faith, and faith has been a rock for me to stand on, especially during the era of COVID-19 and all the challenges and tragedies that has brought to us. So I guess at the moment, I keep coming back to the Psalms when this is such a set of trials. And obviously the Psalms, many of them written by David, are very much about experiencing trials and how do you deal with that. And they're very real. And basically, it's not always like, okay, God, everything's fine here. A lot of the time, it's like, God, why are you doing this to me? But Psalm 46 is a regular one that comes back to my mind, especially in these last 2 years, that God is our refuge and strength, an ever-present help in trouble. Isn't that interesting? So that says you're going to have trouble. Anybody who's going to say, oh, well, just have faith and everything will go smoothly. We all know that's not true. But when things don't go smoothly, God is there as a ever-present help in that trouble. I have leaned on that for my whole life, maybe especially in the last 2.5 years of COVID.
Absolutely.
How about you? What's your favorite verse? I got to go back to you on this one.
I waver on this, but I think right now with some of the things that is happening in my personal life, it is honor thy mother and father.
Okay.
Yeah.
One of those commandments, yes, that we should all think about and always adhere to. Uh-huh.
All right. Well, I truly appreciate you having this conversation and mini church session with me. I wish you the best, and
should we sing or something?
we should have had a choir. I wish you the best, and I am so happy to know you. And of course, congratulations on this award and the plethora of awards that you will continue to get. As your career has certainly been a cornerstone of genomics, but just science in general. And blessings. Have a great weekend.
Thanks, Kizzmekia. And thanks for agreeing to have your arm twisted and to do this
it
interview.
took nothing.
It's a great honor.
I was delighted.